As healthcare technology advances, nanotechnology has emerged as an innovative new way of effective drug delivery. Today we'll be examining the potential, applications, and limitations of this new technology. Drug delivery is the technology utilized to transport the drug to the desired area in the body for drug release and absorption. The delivery system is a formulation or device that enables the introduction of a therapeutic substance into the body and improves its efficacy and safety by controlling the rate, time, and place of release of drugs into the body. Drug delivery technology offers significant benefits to medications such as the extension of the patient's life, a reduction in the side effects, and dosing frequency. In drug delivery, nanoparticle shows great promise by altering the bioavailability, pharmacokinetic, and pharmacodynamics properties of drug molecules in the delivery of the medication. This is possible due to their size, as nanoscale particles are able to alter the chemical and biological properties of molecules. They have found widespread use in many industries, with different production techniques creating a wide variety of different nanoparticle sizes. There has been much attention in nanoparticles in drug delivery over the past decade, and the three major applications we will be examining today are for cancer, oral, and brain drug delivery. In the treatment of cancer, nanoparticles enable much earlier detection and much earlier treatment. The tumor tissue's leaky vasculature causes circulating nanoparticles to accumulate more in tumor tissues than in normal tissues. In addition to this, tumor tissue's drained lymphatic system impedes nanoparticles excretion, further enhancing nanoparticle accumulation within the tumor tissues. Negative surface charge and high drug loading efficiency are the optimal characteristics of this application. Another type of drug delivery made possible through nanotechnology is oral delivery. Due to the poor solubility and stability of traditional oral delivery systems, it is hard to achieve high levels of efficiency. Nanoparticle drug carriers can shield drugs from degradation and deliver them to the intended sites within the gastrointestinal tract, enable more efficient and sustained drug delivery. Finally, the blood-brain barrier is a highly complex structure that protects the central nervous system. Thus, small molecules and macromolecules are insufficient to pass this layer. Nanoparticles are therefore perfect for this application, resulting in a significant increase in the therapeutic efficiency. Now we must delve into the engineering principle and designs that allow nanoparticles to be utilized in these drug delivery applications. Many nanoparticles are intravenously injected and must outsmart several underlying processes taking place within the body. There are several barriers the particles need to overcome before reaching the target site, such as filtration in the kidneys, liver, lungs, and advection in tumor tissues. When designing nanoparticles for effective drug delivery through this highly complex biological system, a focus on the interplay and interaction between the individual cells, individual organs, and the overall connectivity of each organ is extremely important. These interactions can include processes such as cellular uptake, drug dissociation from the nanoparticle, and target organelle interactions. To best understand these interactions, theoretical methods require understanding of key features of the nanoparticle, for example, the size, shape, hydrophobicity, and the interactions with cell membranes, as well as a modeling method that describes cell nanoparticle interactions. By defining these parameters and simulating these interactions, it is possible to draw general conclusions and model the potential effects of any drug delivery accurately. While there are many useful applications for nanoparticles in drug delivery today, there is still a way to go in improving the technology. Several key limitations still exist. The first, which we will explore, is the potential toxicity of nanoparticles in the body. Because of the many different biological microenvironments a nanoparticle interacts with, there is a possibility of toxicity to arise at the molecular, cellular, and tissue level. Each can be extremely detrimental to the health of the patient, and therefore it is necessary to develop nanoparticle formulations that significantly reduce these side effects by controlling drug release depending on the environment. This can be done by increasing drug loading capacity of the nanoparticles, as well as modifying surface characteristics of drug carriers to control drug delivery. Another limitation are the complications that can arise due to the interactions of nanoparticles as they enter the body. Upon entering, nanoparticles instantaneously interact with the biological environment. The presence of a protein corona can drastically change nanoparticle properties such as the shape, size, and charge. In addition to changes in nanoparticle characteristics, endogenous biomolecules that are exposed to the nanoparticle interface may also undergo structural and functional alterations. This could potentially greatly increase cellular uptake and reduce the efficiency and efficacy of the drug delivery system. The final major limitation that needs to be overcome for widespread nanoparticle drug delivery to be possible is the difficulty in testing nanoparticles with accuracy outside the body. Nano-sized objects can aggregate sediment and display different diffusion dynamics. Therefore, nanoparticles may be unsuitable for test conditions, or they may interfere with test results. It can be challenging to correlate in vitro nanoparticle exposure doses to in vivo conditions because of the use of inappropriately high concentrations that are not achievable in vivo, thereby giving a false impression of potential toxicity. One approach is to improve in vitro toxicology assays using 3D cultures, dynamic flow models that can be implemented to give a more realistic depiction of nanoparticle interactions with endothelial cells. Therefore, 3D cultures can give us a better understanding of these mechanics. In summary, there are many current applications of nanoparticles in drug delivery, and the future is promising given the advantageous characteristics of nanoparticles. However, we must be cautious and not overly optimistic of this new technology, as many limitations still exist, including the potential toxicity, the complexity of the human biological environment, modified nano-sized drug carriers, and the difficulty in accurately testing in vitro conditions. If these limitations are able to be conquered, then the applications for nanoparticles for drug delivery can be revolutionary for healthcare.